As we know that the base of evolution is change in DNA, and it is bringing the changes in sequence of amino acids, and it's developing different types of proteins, and these proteins are responsible for different variations. So the major mechanism which is behind these new genetic material is generated during molecular evolution, that is gene duplication. So duplication of a region of DNA that contains a gene, it is playing an important role in formation of new variations in the same trait. This gene duplication is product of several types of errors in DNA replication and in repair machinery. So we are familiar with the process of DNA replication, और हमें मालूम है कि at different steps there is involvement of different enzymes which are playing different roles. और उन enzymes की जो performance है उसके result में हमारे पास एक double copy of DNA prepare होती है at the time of mitosis and meiosis. So अगर इस machinery में जो कि इसकी replicating machinery है उसमें कोई error आ जाती है और resultantly एक duplicate के बजाय 2 duplicates prepare हो जाते हैं. So it is happening, but it is random. It might happen like once in a while, but mostly जो एक repair machinery है वो उसको repair कर देती है. It removes the duplicated gene. But in case ये duplicated gene is left and it is a part of those gametes, and it is there is a fertilization, formation of a new offspring with a duplicated gene, it means there is formation of a new variation. So in this image, we can understand this process of duplication. This is a gene which is being replicated. So instead of preparing one copy of this gene, 2 copies of genes are prepared through the process of replication. So now expression of this duplicated gene, it would be different from this gene. तो इसका रिजल्ट ये निकलेगा कि वो कैरेक्टर जिसको ये जीन रेगुलेट कर रही है, फिनोटाइप में उसका इंप्रेशन अपनी ओरिजिनल जीन से डिफरेंट होगा. सो दैट्स हाउ वेरिएशंस दे अकर इन द वन पॉपुलेशन. जीन डुप्लीकेशन इट कैन अकर बाय इन्वॉल्विंग डिफरेंट टाइप्स ऑफ प्रोसेसेस. उनमें से कुछ एग्जांपल्स, वन इज द एन्यूप्लॉइडी. Aneuploidy is non-disjunction. We know that during the process of meiosis, chromosomes are replicated and they are joined with each other. और जब ये separate होते हैं एक दूसरे से, अगर उस stage के ऊपर 2 chromosomes एक दूसरे से separate ना हो पाएं, which is called the non-disjunction. So resultantly, one cell is getting 2 copies of these chromosomes and one is getting nothing. Similarly, in the case of polyploidy, there is possibility of getting multiple copies more than, as a product of non-disjunction, additional copies of chromosomes will be achieved by the product or the daughter cells. To understand this process of aneuploidy and polyploidy, we watch this video.
Homologous chromosomes fail to separate, then one cell is produced with both homologs, and the other cell has no copies of this chromosome. When the cell divides during meiosis II, the centromeres of both sister chromatids split and they segregate to each of the 2 progeny cells. These 2 cells are unusual in that they contain 2 copies of this chromosome instead of 1. In the other 2 cells produced, there are no copies of this chromosome instead of the usual 1. Upon fusion with a normal haploid gamete, all of the cells produced are abnormal for this chromosome. Half of the cells will have 3 copies of this chromosome instead of the normal 2. These cells are said to be trisomic for this chromosome, and the condition is called trisomy. The other half of the cells will have only 1 copy of this chromosome. These cells are said to be monosomic for this chromosome, and the condition is called monosomy. Let's observe what happens when non-disjunction occurs during the second meiotic division. Homologous chromosomes separate normally during meiosis I, producing 2 cells, each having a pair of sister chromatids. If the sister chromatids in one of the cells fail to separate during meiosis II, then 2 abnormal cells are produced, one having 2 copies of this chromosome and the other having none. Normal chromosome segregation occurs in the other cell, giving rise to 2 normal haploid cells. When these cells fuse with a normal haploid gamete, 2 of the cells produced have the normal set of chromosomes and produce diploid cells. The other 2 cells formed are abnormal, one having 3 copies of this chromosome, trisomic, and one having only 1 copy, monosomic.
So that's how, because of non-disjunction, we can have multiple copies of chromosomes, or we might get like lesser copies of chromosomes as expected. So polyploidy, aneuploidy, दोनों cases में gene duplication जो है वो appear हो सकती है. That is the result of these 2 processes. इसके अलावा the other process is the replication slippage. Replication slippage means during the process of replication, there is an error in that at any stage of this replication, जिसके result में instead of replicating 1 gene, 2 genes sequences are replicated and added in the daughter chromosome. So other than these 3 processes, there are other processes of mutations as well, which haven't been discussed here because this is not a topic about the mutations, it's about this gene duplication. So that's how this gene duplication का result ये निकलता है कि जो उसको holding cells हैं, उस holding organism है, it would show a different phenotype which might be slightly or which may be prominently different from its parents' organisms. So this is one error. It is not being selected by the natural selection, or neither it is another kind of artificial selection. It is just happening randomly, and it is bringing variations because there is possibility that ये offsprings या ये individuals, they might be able to fertilize or might be reproductively active, and they can continue their future generation, and it might lead to formation of new subspecies or maybe a new species in future.